So in a couple of seconds, we'll be live on YouTube. So I guess we'll stay quiet until it's ready. And then, I'll start introducing you around 1 p.m.
Okay, so what is the new link? Hi, Paul. Hello.
Hey, everyone.
Hey, Paul. Just for you to know, we are live on YouTube. And so everything is already broadcasted for friends and family outside of Virginia Tech. And then we'll wait a couple more minutes and, admit everybody into the Zoom room. And I'll introduce Morgan before we get started.
And just since I haven't done a master's exam in biochemistry, is there like a public part and then a private part? So for the most part, the faculty reserve the questions for the private section?
Absolutely. We will start with a public presentation, and at the end of which we'll have questions from the audience. And then we'll move on to a closed door session where Morgan will be defending her thesis in front of a committee.
Perfect, thanks.
All right. All right, well, good afternoon, everybody. It's 1 p.m., so let's get started. Welcome, everybody. My name is Clément Pinochet. I'm an assistant professor in the Department of Biochemistry at Virginia Tech. And it is my great pleasure today to introduce Morgen VanderGiessen, who's about to defend her master's thesis in biochemistry from the biochemistry program here at Virginia Tech. Her master's thesis committee is composed of Dr. Chloé Laronder, assistant professor in the Department of Biochemistry, who is also co-chairing the committee, Dr. Paul Garlier, professor in the Department of Biochemistry, and myself. And Morgan joined my laboratory as an undergraduate researcher in January 2018, and she graduated in spring 2019. And during that time, Morgan second authored a publication in the peer-reviewed journal Insects. She presented her work at the Dennis Lead Undergraduate Research Symposium and at the meeting of the American Chemical Society. Working with Diane Eilertz, a PhD student in the lab, she also co-received a mentoring fellowship from the Department of Biochemistry. And then after graduating in 2019, she worked as a field and lab tech over the summer 2019 before joining the biochemistry graduate program as a master's student, co-advised by Dr. Laronder and myself. Her master's research project involved field-based studies on mosquito ecology that were funded by the Global Change Center and in collaboration with Dr. Luis Escobar from the Department of Fish and Wildlife. But she also conducted lab-based investigations of mosquito chemical ecology. Her work was very well received when she presented it at the meetings of the Entomological Society of America and at the meeting of the Virginia Mosquito Control Association. Morgan supervised 2 undergraduate students, received a travel award from the graduate school at Virginia Tech and a Bruce M. Anderson Award given by the Department of Biochemistry to outstanding second-year graduate students. She has 2 extensive manuscripts in preparation, and she submitted grant proposals to the DoD and the NSF. And this spring, she served as an instructor for Biochem 4116, in addition to being recently accepted to the Infectious Diseases Interdisciplinary Graduate Program at Virginia Tech, where she'll be soon starting her next step of her career as a PhD student. Morgen will give us a 45 to 50-minute presentation that will be followed by questions from the audience here on Zoom, but also in the comments on YouTube, as this defense is broadcasted live on this platform. And then the seminar will be followed by a closed-door defense with our master's committee. So without any further ado, please welcome Morgen VanderGiessen.
Thank you very much for that kind introduction. So I will begin sharing my screen. Can everyone see that all right? Hear me all right? Everything's good? Great. Thank you so much for the thumbs up. So hi, my name is Morgen VanderGiessen, and I will be presenting my thesis on the effects of habitat and plant volatiles on mosquito spatial, seasonal, and chemical ecology. So mosquitoes are the deadliest organism in the world, killing over 1 million people. And sorry, I think there's something. Okay, sorry, I saw a lag on my end. So mosquitoes are the deadliest organism in the world, killing over 1 million people per year due to the pathogens they transmit. And mosquitoes use several sensory modalities to locate necessary nutrients, including plants, which are required for plant nectar and metabolic energy, as well as hosts for blood and reproductive efforts. The most important of these senses is their sense of olfaction, also known as their sense of smell. Hundreds of chemicals which drive mosquito behavior have been identified, and often these chemicals are behaviorally specific for either mating, opposition, sugar feeding, or host seeking. Overall, the majority of semiochemicals which have been identified are related to host seeking, as this is the action that leads to disease transmission. In contrast, the role of sugar feeding in mosquitoes has been relatively overlooked, despite it being a crucial aspect of mosquito biology for both male and female mosquitoes. Furthermore, the olfactory reception and response of mosquitoes to these chemicals is situationally and ecologically dependent. For example, several environmental parameters, including humidity, temperature, the time of day, and sugar feeding status, can influence mosquito behavior and responses to these odors. Here, for an example, consider an olfactory-rich environment, like walking across the Virginia Tech drill field at lunchtime. There are a plethora of odors from ornamental flowers, the smell of coffee and food from dining halls, and even some floral chemicals emitted by personal care products that we use every day. The effects of all these volatiles on mosquitoes and how habitat factors influence mosquito behaviors is relatively unknown. So here we sought out to understand not only what species of mosquitoes are present in ecologically diverse areas, but how mosquitoes handle all of these olfactory signals. So there are a variety of ecological factors which could be influencing mosquito survival and behavior. These include host prevalence, host preference, and environmental characteristics such as temperature, humidity, and plant availability. So therefore, we sought out to address these unknowns by looking at both the habitat and chemical ecology and their effects on mosquito behavior. First, to understand how mosquito habitat and spatial factors influence mosquito prevalence, we performed mosquito trapping and urban population modeling. This allowed us to better understand how habitat-specific climatic conditions drive mosquito prevalence and sugar feeding. Next, since we know that mosquitoes require plant nectar for survival, we became interested in how plant odors influence mosquito behavior. This actually raised an interesting question, considering that humans wear a vast amount of plant-derived chemicals on our skin, and how that impacts mosquitoes is relatively unstudied. Therefore, we sought to investigate how floral volatiles found in personal care products influence mosquito-host interactions. So the first chapter is on the sugar feeding and the species abundance of mosquitoes along landscape gradients. And this project is a collaborative effort between the Lejander Lab, the Vineget Lab in the Department of Biochemistry, and the Escobar Lab in the Department of Fish and Wildlife Conservation. So as mentioned, the role of sugar feeding and environmental and ecological factors which drive mosquito sugar feeding has been relatively overlooked. Several environmental parameters which influence mosquito survival, most notably temperature and humidity, also influence mosquito sugar feeding. And this is important because males exclusively feed on sugar, and therefore without sugar meals, they will die. Similarly, for female mosquitoes, although they can survive on blood meals, sugar consumption is very important for metabolic energy and finding mates and producing eggs. Recently, sugar feeding has been shown to increase when environmental conditions are dry and hot, but the exact conditions and seasonal effects which drive these interactions are unknown. Similarly, high humidity can increase the duration between sugar and blood meals because it reduces the risk of mosquito desiccation. Higher temperature, on the other hand, can accelerate mosquito development and activity. Therefore, these environmental parameters and additional parameters such as precipitation, time of day, and the time of year can directly affect mosquito survival and prevalence. Thus, we hypothesized that the prevalence of sugar feeding and mosquito species abundance is correlated with habitat-specific environmental parameters, especially when looking at diverse urban and forested habitats. So to investigate this hypothesis, we first looked at how environmental parameters shape mosquito species prevalence, and next how the species prevalence of sugar feeding varies between species and habitats. So first, to assess mosquito populations and species diversity, we wanted to choose a wide variety of trapping sites and compare these habitat parameters. So Blacksburg, Virginia is actually an ideal study site due to its close proximity of urban, subforested, and forested environments, as well as a steep elevation gradient. The range of elevations is important as altitude is a great estimator for larger latitudinal ecological factors. So in other words, by using a range of elevations, we can investigate larger geographical features while using a relatively small study site. Therefore, we chose 3 trapping sites, including an urban location in downtown Blacksburg, Virginia, a low elevation forested location at Pandapis Pond, which is just along 460 West on the outskirts of Blacksburg, and a high elevation forested located at Mountain Lake Biological Station in Pembroke, Virginia, which is located on the edge of the National Forest, or Jefferson National Forest, further down 460 West. We would like to thank our collaborators in the Escobar Lab, including Mariana Castenda Guzman and Dr. Luis Escobar, for working with us to assess ecological diversity between the 3 trapping sites. So here we use a variety of satellite-derived data to investigate landscape features such as elevation, vegetation, land surface temperature, and landscape disturbance. So this figure indicates the latitude on the y-axis and the longitude on the x-axis of the 10 different mosquito traps deployed in the 3 ecological habitats. So here you can see that the elevation indicated by the color gradient, which is perhaps one of the most important distinguishing and important factors of these trapping sites. The elevation increases between downtown Blacksburg, the Pandapis Pond, and the Mountain Lake Biological Station. Furthermore, we also investigated the vegetation at these sites, looking at the same X and Y axis, with the green being more vegetation and the red being less vegetation. We also see differing amounts of vegetation between the 3 environments, with the lowest vegetation being in the urban environment. Similarly, when we look at the landscape disturbance on the bottom left, also referred to as nightlight, which reveals light pollution, and the brighter colors indicate more light pollution, with the greatest amount of light pollution occurring in the urban environments, and moderate light pollution in the low elevation forested. Lastly, we looked at the land surface temperature, which is on the bottom right-hand side, where the red color indicates a high land surface temperature, and the land surface temperature is significantly higher in the urban environments. As both of these are proxy for urbanization, you can begin to see that 3 sites are in fact very diverse. And this overall helped us prove that these 3 ecologically diverse environments were distinguished by elevation and urbanization factors. In these environments, we place these mosquito traps, and this is an example of one of our mosquito traps. It is a BG Sentinel trap, and these traps were employed with both CO2 and an odor blend in order to attract mosquitoes. It also has a battery which runs a fan on the interior of the trap, and this fan creates a convection current, which not only creates current that sends the odor plume and CO2 signals away from the trap, but also creates an inward current which traps the mosquitoes in a little mesh bag inside of the trap once they enter the trap opening. Additionally, we put an eye button on the edge of the trap to collect temperature and humidity data. And these traps were deployed for about 16 to 17 hours to allow for the collection of a wide variety of mosquito with different periods of activity, while still maintaining a short enough duration where the mosquitoes were trapped would not fully digest their gut sugars or desiccate in warmer temperatures. So all 1,889 mosquitoes got the full experience, in which from their little trapping bag, they were brought back on dry ice and driven to Virginia Tech. So first they were placed in negative 80 degrees freezer to avoid further digestion of sugar meals and avoid desiccation. Then they were identified based on morphological traits. And then we measured the wing length and the wing size, and then we performed sugar analysis on each of these mosquitoes. So we used a taxonomic key titled The Identification and Geographical Distribution of the Mosquitoes in North America, north of Mexico, written in 2016. And what we see is that a variety of mosquitoes were identified, with some species being present in only the urban or forest environments, while others were identified in both environments. Additionally, an important thing to note is that 2 of these mosquito species are actually invasive to the United States, including Aedes japonicus and Aedes albopictus. Moving into the results, here we show the species of mosquito on the y-axis and the trapping date on the x-axis, divided by the week. So the colors are representative of their trapping environment, where the lighter the color, the fewer mosquitoes were trapped, and the darkening of the color indicates that the more mosquitoes were trapped, with an upper limit of about 150 mosquitoes in the full black. When looking at the time frames of the mosquito trapping, in both the forested environments, we see more mosquitoes trapped in the late summer and early fall, whereas in the urban environments, we see mosquitoes trapped over a longer range from about June to October. This distribution could be driven by species factors, as both forested environments consist primarily of Culex pipiens, and the urban environments primarily of Aedes albopictus. These species also have very different host preferences, which likely contributes to their ecological habitat preference. And so these host preferences are for Culex pipiens, they prefer birds, that's their primary host. They also can feed on mammals and humans. And then for the urban environments, Aedes albopictus, and these mosquitoes prefer feeding on humans and often sometimes mammals such as domestic pets. So then we want to look at the temperature and humidity differences between these 3 different environments. So here, looking first at figures B and C, the temperature and humidity are on the y-axis and the date is on the x-axis. And the shading on the plot indicates where the top is the maximum and the shading on the bottom indicates the minimum, where the center line is going to be the mean. So what we saw is that overall, the urban environment had the highest average temperature and the lowest average humidity. Whereas in the high elevation forested, we saw the lowest average temperature and the highest average humidity, with the low elevation forested being somewhere in the middle. So the temperature and humidity also influence the mosquito trapping in the 3 environments. In figure D, the average humidity is on the y-axis and the average temperature on the x-axis, with the darkening of the colors again indicating a higher number of mosquitoes trapped. In the 2 forested environments, the maximum number of mosquitoes were trapped when the humidity was above about 70%. But they had a higher number of mosquitoes at a larger range of temperatures. In the urban environments, the majority of mosquitoes are trapped over 17 degrees Celsius, and this likely indicates that some mosquito species in these environments have different temperature and humidity tolerances, which makes sense because at higher elevation, where the nightly temperatures get colder, to live in these environments, mosquito species must be able to survive at these nightly lows. Therefore, to investigate this temperature at a species level, we plotted the minimum nightly temperature against the density of the number of mosquitoes trapped at each trapping site. So species such as Culex pipiens and Culex restaurans along the bottom, and Aedes triceriatus and Anopheles punctipennis in the center were trapped more often at lower temperatures indicated by the dashed line threshold, whereas urban mosquito species such as Aedes albopictus and Aedes japonicus in the top row were almost found exclusively when nightly temperatures were well above the minimum temperature. Therefore, we can conclude that species abundance is dependent on both the trapping site and the minimum temperature, and these are both environmentally dependent. So overall, we were able to use the species identity, the trapping date, the temperature, and the humidity to determine how habitat conditions drive mosquito diversity. And overall, we were able to conclude that habitat-specific temperature and humidity conditions drive mosquito species assemblage and abundance. So next, we wanted to assess the sugar feeding of these different mosquito populations. And to do so, we performed an anthrone test, which has both qualitative and quantitative stuff. So the first that is displayed here is the qualitative test, and this can detect whether a mosquito has recently fed on plant sugar. In this instance, we crush a mosquito into 3 milliliters of anthrone, wait 30 minutes, and observe the color change of the solution. A yellow color indicates the mosquito has not fed on sugar, and the blue-green solution indicates the mosquito has recently fed on sugar. This occurs because fructose reacts with anthrone at cold or room temperature, but we can investigate further using a warm anthrone method to determine the total sugar consumption of the mosquito, such as the presence of sugars saved as glycogen by the mosquito. So next, we add 2 milliliters of anthrone to the same test tube, heat the solution, cool the solution, and then determine the absorbance using a spectrophotometer. So the absorbance is then converted to a concentration using a standard curve. So looking at the results of the cold anthrone tests, here we investigate the proportion of sugar-fed mosquitoes on the y-axis and the date on the x-axis, where the colors indicate the environment, with the green being the mountain lake biological station, the high elevation, the Pandapus being the low elevation forested, and then the urban environments. So here, the size of the dot indicates the number of mosquitoes, and we recognize that the greatest proportion of sugar-fed mosquitoes across all environments occurs in June, where the proportion sugar-fed is almost up to 100% or at 100% in the forested mountain lake biological station. Interestingly, this trend occurs in all three environments, at which the lower the number of mosquitoes trapped, the higher the proportion sugar-fed mosquitoes. Furthermore, when looking at the total proportion of sugar-fed mosquitoes per environment in Figure C, we see that we have different numbers of mosquitoes trapped overall. The highest proportion of sugar feeding were trapped in the high elevation forested and the least in the low elevation forested. When looking at the actual species which drive this interaction, knowing that the urban environment consists mostly of Aedes albopictus mosquitoes, we see that these mosquitoes has a relatively low percentage of sugar-fed mosquitoes at about 14% sugar-fed. But Culex mosquitoes in the forested environments have an even lower sugar-fed population, which is about 5.6% sugar-fed. So to further characterize the influence of the number of mosquitoes trapped on the sugar feeding, we looked at the proportion of sugar-fed mosquitoes versus the number of mosquitoes trapped. So here we see similar results, in which the fewer the number of mosquitoes, most prominently in the forested mountain lake biological station site, the higher the proportion of sugar feeding. Whereas in the urban and forested Pandapus site, as the number of mosquitoes increases, the fewer mosquitoes have sugar-fed. So therefore, we can conclude that the proportion of sugar-fed mosquitoes is mediated by both the time of the year or the date and the habitat type. So the sugar feeding ability is impacted by mosquito body size. So next we looked at how the mosquito body size could influence sugar feeding. So looking at exactly how the mosquito body size influences the total carbohydrate content intake of mosquitoes. So previous research has indicated conflicting results on the relationship of sugar feeding and body size. In some studies, they say the body size of mosquitoes correlates with the amount of sugar a mosquito's intake, yet others have not found the relationship between these two. So here we used a micrometer under a microscope to measure the mosquito wing along the top subcostal vein in order to estimate body size. So in Figure D, we took the log of the carbohydrate content from the worn anthrone test on the y-axis and plotted it against the mosquito wing size on the x-axis. So the X's are the mosquitoes that were trapped that were not sugar-fed in gray. And the triangles in gold are the sugar-fed mosquitoes from the cold anthrone test. So here we see a significant relationship between both the length of mosquitoes which have sugar-fed and the mosquitoes which have not sugar-fed to the log of the total carbohydrates. So we wanted to assess whether this was species specific, as different mosquito species have relatively different body sizes. And these graphs contain the same X and Y axis and the same sugar-fed and non-sugar-fed groups for both Aedes albopictus mosquitoes in the urban environments and Culex pipiens in the forested environments. And what we see is that the mosquito wing sizes do not correlate with the total carbohydrate of sugar-fed Culex pipiens and Aedes albopictus. But interestingly, they do correlate with Aedes albopictus mosquitoes which have not been sugar-fed. Therefore, we were able to conclude that the total carbohydrate intake correlates with mosquito body size, but the correlation may be influenced by sample size or the individual mosquito species. So together, this data has allowed us to assess a variety of parameters and determine how mosquito prevalence and sugar feeding is related to ecological environment. Specifically, we identified that Culex pipiens and Aedes albopictus were the most prevalent species in their respective environments. So next, we wanted to assess whether these environmental variables could actually predict the urban mosquito species Aedes albopictus. So to do this, we used a generalized linear model to predict Aedes albopictus using the minimum, the mean, and the maximum temperature and humidity values. Additionally, we generated a variety of models by testing different combinations of these variables in order to compare different models and find the best fit for our dataset. To do so, we compared these models using an Hike information criterion, which is going to be future referred to as AIC. This is just a statistical tool which allows us to estimate and compare the quality of each model. So first, in this figure, we compare the inclusion and exclusion of different variables using the AIC value. So here what we're looking at is the AIC value on the left-hand side, and the lower the AIC, the better the model is at predicting mosquito populations. And again, these models are just different combinations of variables. So for example, a less accurate model would be located towards the top, and a more accurate model would be located towards the bottom. Using all of the variables collected, indicated by the solid pink arrow, we were able to get a relatively good model, which I'll show you graphically in a minute. But there are models which exclude additional variables that actually do a better job predicting the number of mosquitoes, indicated by the dashed arrow. So now looking at the generalized linear model plot, you can see the actual field captures in black with the date on the x-axis and the number of mosquitoes on the y-axis. The full GLM model closely resembles the field captures, but also take a look at the minimal GLM model, again indicated by the dashed lines. This model also closely resembles the data, and as indicated by the AAC value, actually better predicts the number of field captured mosquitoes. Therefore, this indicates the three most important predictors for Aedes albopictus populations are actually the mean temperature, the minimum temperature, and the mean relative humidity. Therefore, we can confirm that we can predict Aedes albopictus populations using these three values and overall using the temperature and humidity values. So what we found is that sugar feeding and species prevalence is largely dependent on habitat-specific environmental parameters, and some of this also may be driven by host preference and cold tolerance, which drive mosquitoes to live in a particular habitat. We also identified an inverse relationship between the number of mosquitoes and the proportion of sugar-fed mosquitoes. We also predict that habitat-specific vegetation potentially drives mosquito sugar feeding and mosquito prevalence, seeing as the most sugar-feeding mosquitoes are trapped in the high elevation forested with a moderate amount urban and the lowest in the low elevation forested location, despite some overlapping species between these environments. And we also identified that we can model our urban mosquito populations using temperature and humidity. In addition, we actually tried to model the Culex pipiens mosquitoes using the temperature and humidity values, but were unsuccessful. Therefore, it's potentially something more complex about these mosquito biology, which influences their prevalence and survival in forested habitats. Future studies could also expand this work by characterizing the plant composition in the three different ecosystems, including the sugar accessibility, the plant type, and the olfactory profile of these plants. This could also be used to further understand mosquito survival in a habitat-dependent context. As demonstrated, we can predict these Aedes albopictus mosquitoes using just the temperature and humidity values. But for Culex mosquitoes, the temperature and humidity are not sufficient. So potentially future studies could investigate precipitation or the prevalence of other growth stages. For example, we could observe the prevalence of Culex egg rafts, larvae, and pupae in nearby water sources to determine whether overwintering is causing weaker mosquitoes to die when either the temperatures get very low or just living in less than ideal conditions. Additionally, this work can be crucial for investigating how mosquito populations change with predicted environmental changes, such as increasing temperature or humidity based on climate change, and how these impact mosquito populations, especially in urban environments. So shifting gears a little bit now that we know that mosquito species assemblage and plants are relatively diverse across different habitats, how does the plant diversity and therefore floral volatiles influence mosquito behavior? So as previously mentioned, mosquitoes locate both blood and nectar sources based on their sense of olfaction. And floral volatile differences between wildflowers and ornamental flowers, such as those planted in urban areas, could significantly influence mosquito habitation and survival. But these plant volatiles are not actually just limited to plants. As we wash ourselves with scented soaps and shampoos, even wash our clothes with detergent that's scented with plant-derived chemicals, some of which we actually know are detected by mosquitoes. So how mosquitoes deal with the combination of these host volatiles from our skin and the combined personal care products is relatively unknown. Therefore, in Chapter 2, titled The Effects of Personal Health Products on Mosquito-Host Interactions, we investigate how personal care products, specifically body washes, influence human odors and the resulting mosquito host-seeking behaviors. So to investigate how personal health products influence mosquito host seeking, we use Aedes aegypti mosquitoes, and they prefer urban habitats and are anthrophilic mosquitoes, which means they almost exclusively feed on humans. Several singular and blends of chemicals have been identified as olfactory cues for both nectar and host-seeking behaviors of these mosquitoes. Additionally, some research has been done on the influence of odor blends on Aedes aegypti mosquitoes. One example is that blends of both host-associated compounds and plant sugars have previously been proven to be attractive to these mosquitoes in the context of mosquito trapping and baiting. Some components of personal care products have also been investigated to impact these mosquitoes, including essential oils, which can either repel or attract these mosquitoes dependent on both the identity of the oil and the concentration. In addition, some research has been done on the malaria mosquito, Anopheles gambiae, and that has revealed that some antiperspirant chemicals and deodorant chemicals can repel mosquitoes. But overall, it's relatively unknown how mosquitoes respond to the combination of plant-derived volatiles and chemical human odors. So here we hypothesize that soap application adds floral volatiles to the headspace composition of human skin, and this increases mosquito attraction. So to address this hypothesis, we first investigated how personal care products contribute to our overall odor profile, and next investigate how these odors influence mosquito attraction. So we chose 4 different soaps here. So the first 2 are Dove and Dial soaps, and then there's scent, or there's specific one that we picked out next to them. So 114 million Americans used Dove in 2019, and 58 million Americans used Dial in 2019. So this makes up about 50% of the population of the United States. The other 2 soaps that we chose is Simple Truth, which is a Kroger brand organic soap. And then we also chose a Native brand, which is an emerging brand in popularity, which is known for having minimal ingredients and also being organic. So while we chose some of these based on popularity, we also chose them based on their ingredients. So some of them contain coconut oil derivatives and plant extracts. And it's important to note that coconut oil has been recently demonstrated as a stronger repellent than the gold standard DEET to blood-sucking insects, including mosquitoes. So to sample each individual's odor, we washed nylon sleeves and cut them into we cut 1-inch sections of nylon sleeves, and we washed them in a scent-free detergent. After they were given time to dry, we performed odor collection of individuals' arms. So first we washed both arms with DI water to remove any contaminants. And then we applied 2 control sleeves on one arm and covered that in foil to avoid clothing from touching it or any contamination. Then the other arm we washed with one of the soaps of choice for 10 seconds, and then applied 2 sleeves to that arm and covered in foil and sampled the odor for 1 hour. So we used 2 sleeves on each arm in order to take 1 set, so 1 soap and 1 control, and do chemical analysis of it. And then the other was used to test mosquito behavior in a cage assay. So our odor collection technique was using SPME, so solid phase microextraction, and this is a sensitive fiber which collects volatile and semi-volatile chemicals. So this graphic of the SPME is on the right-hand side. So the SPME fiber is held by a SPME fiber holder, and you can withdraw and extend this fiber in order to expose it to an odor and then retract it to avoid getting external odors. So for the SPME sampling, what we did was we baked the SPME for 3 minutes to clean it. Then we, in a jar, we had already collected the odor from an individual. Then we put the internal standard onto the sleeve in a jar, exposed the fiber to the sample for 1 hour, and then insert into the GCMS for 3 minutes. And the GCMS was used to analyze the chemicals in the sample and the abundance of each chemical again. So here is an image of all of the sample spectra. So here in the first row is the clean arm without the soap of the 4 individuals tested, consisting of 2 males and 2 females. And then below them are the spectra of the individuals' arms after being washed with Dial, Dove, Simple Truth, and Native soaps in that order. So these spectra were scaled to have the retention time of 24 minutes on the the x-axis, which correlates with the chemical compound's molecular weight. And the y-axis is the abundance of the different chemicals. So upon first glance, you can see that each individual has a relatively diverse odor profile, but the overall odor abundance and number of odors indicated by the number and height of the peaks significantly increases when washed with soap. Additionally, we can see that the more manufactured non-organic soaps, Dove and Dial, significantly increase the volatility and the number of compounds, whereas the organic natural soaps, Simple Truth and Native, more closely resemble that of the control odors. So next we wanted to determine the chemicals which were driving this interaction. So what chemicals are actually here? So we suspected a variety of different odors, first being a variety of soap volatiles, so things such as colorants, artificial fragrances, other soap additives such as antiperspirants, things like that, antibacterials. We also expect some host volatiles and components of human skin and human sweat, such as nonanol and octanol, which are known to attract mosquitoes to human hosts. And we also expect some plant volatiles like linalool and limonene and things that can attract or repel mosquitoes from different species of plants. So to visualize the differences and similarities between samples, we used a multivariate analysis called non-metric multidimensional sampling. So what does an NMDS do? So what this does is it takes multidimensional chemical data and ranks each dimension. So consider that there is one chemical that is highly abundant in one sample and not very abundant in another. It's going to rank these based on their differences. And next, it plots these rank orders on a two-dimensional matrix. So the closer the sample, the more similar these two samples are. And the farther the sample is, the less similar those samples are going to be. So we had a total of 69 samples, and the shapes indicate the volunteers. So we had 4 volunteers with 2 male and 2 female, and we had 4 replicates of each. The colors indicate the soap washed conditions. So the 4 soap washed samples, we have 3 replicates of each. And so the gray is going to be the clean arm samples of all of the individuals. The orange is going to be the dial washed samples from all of the individuals. The blue is going to be the Dove washed samples from all the individuals, yellow Simple Truth, and green native washed. So what we can see is that the chemical signatures of the controls are significantly different from the soap washed samples. This is indicated by the gray ellipse being separated from the soap samples. And we also see that the 2 heavily fragrant and non-organic soaps cluster, the Dove and Dial, whereas the 2 more natural soaps also cluster, the Native and the Simple Truth. So since we identified hundreds of chemicals across all of these samples, in order to pare down the data and make conclusions about how soap influences human body odor, we divided these chemicals into their functional groups. So here we display the abundance of each chemical functional group from the average of the sample replicates. So on the y-axis is the abundance of the chemicals, and on the x-axis contains the sample identification. The first 4 here are the clean arm control samples, and as you would expect, each volunteer has an individual chemical odor profile, as we all smell different. But aldehydes in the peach color constitute the majority of all human body odors across all 4 volunteers. When looking at the soap samples, the majority of soaps contain terpenes, which is in the purple color, and very few aldehydes. And so these are also Dove, Dial, Simple Truth, and Native, and this is just the soap only. So when you look at the soap washed samples, and these are the average of all of the soap washed samples regardless of the volunteer, you can see that the soap washed samples closely resemble the soap only samples, and compared to the clean arm samples on the left side. We can also see similar chemical functional group abundances in the soap and the soap only samples compared to the soap samples. Specifically, the clean arm consists on average about 50% aldehydes, again in peach. But the soap wash samples, we only see about 15% aldehydes. On the other hand, in the clean arm samples, we see less than 10% terpenes, also in the purple color. When we took the average of all the soap wash samples, we see about 40% terpenes, which is a significant 30% increase in the amount of terpenes, or the abundance of terpenes across these samples. So therefore, we wanted to further investigate which chemicals were driving these differences. So here is the relative abundance on the y-axis and the average of all the clean arms in the first bar, and then the average of the soap wash samples in the subsequent bars. I selected for chemicals which made greater than 10% of each sample, and the rest of the chemicals which made less than 10% of the sample were filtered into the other category. Additionally, the table on the bottom is the same as the data in the graph, but also has the chemical group added in the first row. So first we can see D-limonene in the lighter green color on the bottom and M-camphorine in the medium blue, also towards the bottom on the graph, are the two most abundant samples in both the Simple Truth and the native soaps. Similarly, the Dove and the dial wash samples consist of D-limonene and linalool, which is in the yellow of the other two chemicals, varying based on the soap samples. So this is interesting because limonene as well as linalool have previously been studied as mosquito repellent compounds. Additionally, while M-camphorine has not been studied against mosquitoes, it has been shown to repel a different species of insects. But again, these repellency effects are highly dependent on concentration. So how do these chemical differences influence mosquito behavior? To do this, we did a behavioral cage assay. First, these mosquitoes were synchronized to a peak host seeking time of Zet-Geber 10 to 12, which is about the evening time. Each of the odors were placed into a cage. So for this example, we're going to say that we put one clean arm and then one soap-washed arm into the cage. Then groups of mosquitoes were released into the cage and recorded for 20 minutes. For this, we did about 2 to 3 replicates per odor set in order to achieve greater than 20 mosquito landings. And then we quantified both the preference of the mosquitoes and the total number of landings. So here, the preference index on the y-axis indicates whether a mosquito preferred the test odor, indicated by a positive number, had no preference to either odor, indicated by 0, or had a preference for the control, indicated by the negative numbers. Additionally, the numbers within the bar are the number of mosquitoes which made a choice because one of the two cups. And along the bottom of the graph is the total number of mosquitoes used in each cage assay and the volunteer identity. So first, we put 2 clean socks into the cage as a control to confirm no bias for either side of the cage, and that's in the black bar. And so we confirmed that there was no bias of the cage. Next, we determined the preference of mosquitoes to each of our volunteers, clean odor. So we did a clean odor, or the clean arm is on the top, indicated by the tan circle, and then the clean sock, indicated by the gray, was on the bottom. So all 4 individuals were preferred by the mosquito compared to the odorless control, with female 1 and male 2 being the most attractive to mosquitoes. Next, we looked at the preference of mosquitoes to the soap wash samples compared to the clean arm sample, with the clean arm samples being again indicated by the tan dot below. And we see that the mosquitoes prefer the individuals washed with the Dial, Dove, and Simple Truth samples compared to the clean arm. But in the case of the native washed soap, these mosquitoes prefer the clean arm over the soap washed arm. So overall, we were able to identify that odor profiles of soap washed samples are mostly driven by the soap usage, only minorly based on human odor variations. We also saw that soaps reduce the percentage of aldehydes and increase the terpene and aromatic volatiles in the samples. There's an overall reduction of host-seeking volatiles used by mosquitoes in the soap samples versus the control. So some of this could be driven that previous studies indicate that mosquitoes can detect aldehydes and ketones, amines, with little or less detection of alcohols or aromatics. So potentially mosquitoes can't actually detect all of these compounds that we tested. Looking at the behavior of these mosquitoes to these soap wash samples, the degree of attraction to the soap wash samples is very dependent on the individual human odor variations. And we saw that Simple Truth, Dove, and Dial soaps had similar behavior to the controls, despite having different odor profiles. And potentially this is because mosquitoes can't detect or are not influenced by some of these terpene and aromatics, which drive the differences between these soap washed samples. Future studies of the influence of personal care products and mosquito behavior could expand the soap and volunteer pool. So here we chose soaps with scents such as marula oil, honey blossom, and coconut, where we saw several plant fragrances such as limonene, linalool, and lilau. And potentially would be informative to look at soaps with other plant fragrances such as lilies or rose-scented soaps. We could also use these findings to better inform personal care product companies which ingredients could be attracting mosquitoes to human beings to avoid their usage in personal care products. Furthermore, we could expand sampling methods and or the odor collection method to control the exercise to determine how, such as human sweat odors induced by exercise influence the odor of personal care product usage, and how human odor is impacted by that. Finally, we could perform single odor behavioral tests to determine what ingredients are causing mosquitoes to have this aversion from these native wash samples. And interestingly, though, although camphorine and D-limonene are the two most abundant compounds in the native soap, they were also present in the Simple Truth soap, and they had some of the same kind of chemicals and similar concentrations. Therefore, we could establish a concentration gradient and determine what concentrations of these chemicals and chemical compounds are providing these repellency effects. So overall, we narrowed two major knowledge gaps on the mosquito species abundance and the effect of personal care products. So first, we were able to determine that seasonal and spatial ecological factors significantly influence mosquito abundance and sugar feeding behaviors, and we can use these to model urban mosquito populations. Also, we noticed that soaps from personal care products significantly influence individual human odor profiles, and these personal care products could be influencing mosquito biting behaviors dependent on their volatile ingredients. So together, we were able to identify that mosquito species biology and habitat and chemical ecology factors are very important to understanding mosquito prevalence and biting behaviors. Specifically, we found that temperature and humidity influence mosquito populations, but other factors are also at play, including habitat-specific host availability, as well as potentially flower prevalence and ornamental and wildflower preferences. Other factors may be species-specific characteristics, such as mosquito cold tolerance, or how they survive winter, whether that be an egg or adult form, and their host preferences, whether that be human, mammalian, or avian. And the most important takeaway from this research is that several things that we are doing on a daily basis and the conditions around us influence mosquito populations. For example, the global climate change could increase the rate or the range of mosquito survival, and increasing urbanization could increase the prevalence of several vector important species, such as Aedes albopictus and Aedes aegypti, and thus increase the chances of vector-borne disease transmission. Additionally, the role of plant volatiles from ornamental flowers and floral home and personal care products could be drawing mosquitoes into urban areas and also increasing the likelihood of mosquito biting events. Therefore, it is crucial to expand our knowledge on how ecological factors influence mosquito prevalence and biting. And with that, I would like to thank my advisors, Dr. Chloe Lejander and Dr. Clement Vinager, for all of their encouragement, advisement, and infectious passion for vector-borne disease research. I will always thank both Dr. Lejander and Dr. Vinager for seeing my potential in me when I joined the lab as an undergrad 3.5 years ago, and also allowing me to pursue a master's degree in their laboratories. I would also like to thank Dr. Parth Collier for his invaluable chemical analysis advice, and members of the Vinager and Lejander labs who participated in mosquito trapping and odor sampling, most notably Ford Upshur for his help trapping mosquitoes, as well as Sarah Tartabini for helping me with sugar analysis in the Lejander lab. And in the Vinager lab, I would like to thank Diana Lertz for odor sampling and method improvements, as well as undergraduate Brenda Miko for aiding with the odor sampling and chemical analysis. I would also again like to thank Dr. Luis Escobar and Mariana Castenda Guzman for aiding with mosquito trapping site investigation in our collaborative project. Furthermore, I would like to thank the biochemistry department for allowing me to pursue this research and being my home for the past 6 years. I'd also like to thank the Global Change Center, the Epley Foundation for Research, the Fralin Life Science Institute, the Center for Emerging Zoonotic and Arthropod-Borne Pathogens for funding. And lastly, I would like to thank my parents for all of their support and encouragement, and I would not be here without them. And with that, I would gladly accept any questions.
All right, thank you, Morgan, for the presentation. Now we'll take some questions from the audience before moving on to the closed door session. And we have a first question in the chat. Sorry, it's a comment. Mariana is congratulating you.
I can pull up the chat. Sorry.
What questions from the audience do we have?
Hey, Morgan. Great job. I have a question. I'm not sure if I just missed it. So sorry if that's the case. It's from your first the first section talking about sugar feeding. I'm wondering why you would catch some or a lower proportion that didn't sugar feed, because don't they all need sugar to survive? Or is it more a product of the environment? They're less dehydrated, so they might eat more, or they might blood feed instead of sugar feeding. What do you think is going on there?
So we believe this is partially based on the mosquito trapping design. So we chose these mosquito traps because they catch a wide variety of mosquitoes. And since we wanted to do both analysis of the species in the area and sugar feeding, we chose these kind of host-seeking traps. But there have been previous papers that have collected mosquitoes using aerators and basically just catching live sitting mosquitoes. And mosquitoes which have sugar fed or recently sugar fed are more likely to be resting, as they can't immediately fly after ingesting sugar meals. So potentially it's just our trapping method estimating a lower sugar feeding population in the area than is actually occurring.
I have a question from Megan in the chat, who's asking, when you did your sugar sampling in the mosquitoes from different environments, did you sample both male and female mosquitoes or just female mosquitoes?
So we sampled both male and female mosquitoes, but the majority of the data presented here is just female mosquitoes, as this is the primary mosquito species that are attracted to these traps, again, because it's a host-seeking trap and males only feed on sugar. And females are the ones doing primarily the host seeking. So we caught a relatively low number of male mosquitoes, so we did not add them into this data.
Any other question from the audience?
Hi, Morgan. I have a question for you. I'm just curious how the source of the sugar that the mosquitoes feed on impacts any aspect of either, I don't know, their lifespan or their vector competence. Is that kind of known, how the source of the food impacts?
Yeah, there's a little bit known about nectar quality can influence the duration between sugar meals, but we didn't look at exactly what they were feeding on. We just wanted to look at whether they had recently fed on sugar or there was some sugar storage here. But yeah, there is a lot of research done on the different kind of plant types, especially for mosquito baiting, how to create these like toxic sugar baits that mosquitoes can feed on and ingest, because some of them are using fruits, and different fruits have different kind of nutritional qualities for the mosquito and can change how long they can go without having another sugar meal.
Have time for another question if you have any pressing question for Morgan.
I have a quick question. So what was, I'm just interested in really interested in the second half of your talk regarding soaps. Do you think you would see sort of similar results? I don't know if this would be feasible or not, but if you use lotion as opposed to soap, because like there's sort of that lotion kind of, I don't know, I feel like the smell stays longer on the skin. So I don't know, do you think there would be any difference between using soap versus lotion? Again, I'm not sure how method-wise how that would work, but I'm just wondering.
Yeah, that's a really interesting. We did not test lotions in this. When choosing body washes, we kind of wanted to use something that the majority of people use, whereas not all people use lotion, and something that's used kind of whole body. So to avoid using shampoos and things that wouldn't be covering the whole body. But yeah, something that might be applicable to that kind of research is that part of like the different mosquitoes choose hosts based on kind of their skin microbes. And that's really different between different individuals. And also your skin porosity could be very different between individuals. So a lotion may be more, may clog the pores more and may actually influence the odor more and more consistently across different groups, is what I would predict. So yeah, that could be really interesting.
Okay, great. Thank you. Great talk.
Thank you.
All right. Well, if there are no other additional questions, let's give another round of applause to Morgan, and then we'll transition to the closed door session of her master's thesis defense.
Thank you. Thanks everyone for coming.
For those of you watching us on YouTube, I'll have to stop the broadcast as the content of the conversation is confidential. Then we'll get you informed of the outcome of the discussion on the comment on YouTube.